Hello students, welcome to Zoology Classroom. In today's class, let us know about sex determination. First of all, let us know what is sex determination. So confirming the male or female gender of an animal is called as sex determination. And how we will determine the sex of an organism. It will be done based on the sexual characters. In case of human beings, we have two types of sexual characters. It is primary sexual characters and secondary sexual characters. And we can see these two types of characters to determine the sex of the organism in case of many higher level animals, especially like birds and mammals. So what are these primary sexual characters? Primary sexual characters are the external genitalia and the gonads of the organisms, like they may be testes or they may be ovaries. So if the male external genitalia and testes are there, they are called as male sexual primary characters. And ovaries and female external genitalia, they make the primary sexual characters of female organisms. And what are secondary sexual characters? Secondary sexual characters can be seen once the organism matures fully in terms of sexual maturity. So in human beings, we can see once the human beings become sexually mature, then we can see secondary sexual characters like developing mammary glands and mustache. So such type of characters are called as secondary sexual characters. So based on these characters, we can determine the sex of the organism. But in case of many lower level organisms, we cannot see any changes externally in terms of secondary sexual characters or primary sexual characters. So in how this sexual determination is done in those organisms. Usually, on the whole, in animal kingdom, there are four mechanisms are there to determine the sex of the individual or organism. They are genetical mechanisms, metabolical mechanisms, hormonal mechanisms, and environmental mechanisms. These are the four different types of mechanisms that control the sexual determination in the organisms. First of all, let us see about genetically controlled sexual determination mechanisms. So in this type, there are again four subtypes are there. Sex chromosome mechanism, it is also called as heterogamesis. Another one is genic balance mechanism. Third one is male haploidy or haplodiploidy mechanism. Fourth one is single gene control of sex mechanism. So first of all, let us see the history of the sex chromosomes, how they were discovered. In 1891, H. Henking was doing some experiments on wasp. So he was studying the spermatogenesis process in these wasps. So during his discoveries or during his studies, he has observed a peculiar nuclear character in the gametes in the sperms of these wasps. And this particular nuclear structure, it occupied almost 50% of the sperm cells. And 50% of sperms were containing this particular structure, and remaining 50% of sperms were not having that structure. So, and he did not speculate on the significance of this structure, which is present in the 50% of the sperms, and he named that object as that nuclear structure as X body. Now, later scientists discovered that that X body is nothing but X chromosome. Later in 1902, C.E. McClung, he also made extensive observations in grasshoppers, and he was also doing experiments or observing about the spermatogenesis process. in these organisms, while doing experiments on the spermatogenesis process, he also found the X body to be present in the half of the organisms, half of the sperm cells. And he determined he suggested that this X body is determining the sex of the uh, embryos. So, and he also reported that somatic cells of female grasshoppers, they contain 24 chromosomes, while somatic cells of the males are having 23 chromosomes. And E.B. Wilson in 1905, he was conducting experiments on females of Proteiner. He observed that 7 pairs of chromosomes in these females, whereas males were having only 6. And apart from 6 paired chromosomes, 1 unpaired chromosomes also was discovered in male Proteiner animals. And he called that unpaired chromosome as the X chromosome. And coming to the structure of X chromosomes, so X and Y chromosomes, they are called as allosomes or sexual chromosomes. And this X chromosome, in many organisms, it is straight in appearance or in structure, looks like a rod-like structure, and it is comparatively longer than Y chromosome. And this X chromosome contains a large euchromatin part, and small amount of heterochromatin will be there. As it is having large amount of euchromatin, large amount of DNA also will be there. And euchromatin consists of a lot of functional DNA, and heterochromatin consists of junk DNA. As it is having a lot of euchromatin, hence much genetic information also will be there. That means many genes will be there in X chromosome. And Y chromosome contains only small amount of euchromatin, large amount of heterochromatin. So what does it mean? Y chromosome contains only a little genetic information. That means only few number of genes will be there. Remaining all DNA is nothing but junk DNA. And that's why sometimes this Y chromosome is referred as genetically it is inert or inactive. And let us see the structure of Y chromosome. So the Y chromosome in many organisms consists of 2 chromatids, single chromatid, 2 arms. Out of these 2 arms, one is the short arm, another one is the long arm. And in this Y chromosome, we can find 2 parts. They are pars and MSYs. Pars are nothing but pseudo-autosomal regions. So these pseudo-autosomal regions, there are 2 pseudo-autosomal regions are there, PAR1 on one arm and on other arm PAR2. So 2 parts are there. And these 2 parts, they are similar to the portions of the X chromosome. So these portions are present on X chromosome also. So these 2 PAR1, PAR2 portions, as they are present on X chromosome also, hence they are called as pseudo-autosomes. And it is comprises of only 5% of the Y chromosome. Remaining 95% of the chromosome portion of Y chromosome is limited to only Y chromosome. So that's why it is called as male specific region, MSR, or male specific Y chromosome region. And in this region only, majority of the genes which are responsible for the male gender will be located in this region. And out of those very important genes, SRI gene and AZF genes, these 2 genes are very, very important for the production of sperms in male organisms. And the sex determinations in male is also determined by this SRY gene on the Y chromosome. So here we can see these 2 X chromosomes in females, 1 X and 1 Y chromosome in the females. So in the embryonic position, these 2 are the embryonic gonads of the male and of the female. So during the early stages of this embryonic development, both these gonads almost appear like same. But due to the SRY gene in this Y chromosome, it determines a factor called as TDF, testis determining factor. So due to this SRY, what happens? Testis development will be done in that gonad. And due to the lack of this Y chromosome portion or SRY gene, TDF will not be developed or TDF will not be produced in the primary gonadal embryonic gonads of the female, so that this type of eggs will be developed into ovaries. If by any chance, if we can control the production of this TDF in this male embryos, these will fail to convert into testes, and they will also develop just like as ovaries. So due to this, we can decide that, or we can say that it is very, very important to make this primary gonad into a testis. What is it is important? Having a TDF factor, and TDF factor will be controlled by this SRY gene. So SRY gene is very, very important for male gonad formation. And in this genetically type, genes are very, very important. Chromosomes are very, very important to determine the sex of the individual. So in this type, two types of mechanisms are there: heterogametic males and heterogametic females. So under heterogametic males category, we can find the XXXO type sex determination, XXXY type determination. So in these two types, males produce two types of gametes. In first type, half of the sperms will be having X chromosome, remaining half will be having no sex chromosome at all. And in XX and XY case, half of the sperms will be having X chromosome and remaining half will be having Y chromosome. So here in this case, the females produce similar type of gametes in terms of sex chromosome. Each ova will be having one X chromosome, so that all X are similar in terms of sex chromosome. But here in males, in these two categories, half males having a X chromosome, remaining half males having either a Y chromosome in the second case or no sex chromosome at all in this first case. And in heterogametic females, females produce two types of eggs, whereas males produce same type of sperms. So here in this thing, ZO, Zed and ZW, ZZ, these two types of systems are there. In these two cases, females produce same type of sperms, all having a Z chromosome as the sex chromosome, whereas females produce two types of eggs. In first case, half X will be having Z chromosome, remaining half will be having no sex chromosome at all. In second case, half of the X will be having Z chromosome, remaining half will be having Y chromosome, W chromosome as the sex chromosome. So let us observe these four types one by one. First one is XX and XO system. This system can be seen in Vallisneria plant and in insects that belong to the orders Hemiptera and Arthroptera. Under Hemiptera, these grasshoppers and wasps will come. And these insects and Vallisneria plants, in these organisms, females, they will have two X chromosomes, whereas males have only one X chromosome. And so only one X chromosome will be there. So males they will produce two types of sperms. Half sperms will have X chromosome, remaining half will not have any sex chromosome at all. So it is the sperms that will decide the gender of the that will determine the sex of the offspring. And in XXXY type, this system can be seen in human beings and insects like Drosophila and some angiospermic plants like Melandrium and Humulus and Coccinia indica. In this, females have two X chromosomes, so they will produce same type of X, all possessing X chromosome. Males possess one X and one Y chromosome, so they will produce two types of sperms. Half will be having X chromosome, remaining half will be having Y chromosome. So it is the sperms that will decide the gender of the, or that will determine the sex of the offspring. So if the Y chromosome bearing sperm unites with this ova, that will develop into a male offspring. If the sperm having X chromosome unites with this ova, that will develop into female offspring. So this is the normal human karyotype of human beings. So in this we can see two X chromosomes are longer than the Y chromosome, So in males, only one X chromosome will be there, which is longer than other one. The other one is Y chromosome. It is shorter than the X chromosome. And sex determination in Drosophila. In Drosophila also, they are also having XX and XY type of method. So in Drosophila, SRY gene will not be there. In human beings, SRY gene will be there to determine the production or development of testis. So in these organisms, apart from SRY gene, this what the ratio of X chromosomes to the set of autosomes determines the sex of these individuals in Drosophila. So if all autosomes are having 2 in each set, then they will be called as normal diploids. If the organisms are having 3 chromosomes, then the autosomal set is said to be 3. So in this way, the ratio of X chromosomes to the sets of autosome determines the sex in these organisms, and that is called the genetic balance system. We will discuss about this a little while later. So let us see about this third system, ZOZZ system. This system can be seen in moths and butterflies. So these organisms, these moths and butterflies, female possesses 1 Z chromosome only, whereas males possess 2 Z chromosomes in them. So here females produce 2 types of gametes. Half having ova will contain this Z chromosome, remaining half will not have any sex chromosome at all. So they are called as heterogametic. Whereas the males produce only 1 type of sperms, all will be having single Z chromosome in them. So the type of egg will decide the gender of the offspring, or it will determine the sex of the offspring. So another last system in this genetic determination methods, ZW and ZZ system. This system can be seen in insects, vertebrates like amphibians, reptiles, birds, and some plants like Fragaris elatior. So in these organisms, male produces 2 types of eggs. Half X will possess Z chromosome, remaining will possess W chromosome. And whereas males produce same type of chromosomes, same type of sperms, all having Z chromosome. So the egg here also, egg only decides the sex of the future offspring. So, and coming to the genetic balance or genic balance system in Drosophila. So in this Drosophila, this system was discovered by Kelvin Bridges. So he was conducting experiments with this Drosophila. So he has obtained some triploid organisms and some tetraploid organisms also in them. So having normal diploids is normal thing, but in rare cases, he was succeeded in getting triploid organisms also. So if triploid is there, all autosomes are 3 in number. If diploid organism is there, it will possess only 2 sets of the chromosomes. And if tetraploids are there, 4 chromosomes will be there. So here in this system, what is happening in this system? The ratio of X chromosomes to the set of the chromosomes decides the sex of the individuals. So this is called gene balance. So X chromosomes number by autosome sets decides the organism's sex in this Drosophila. So this is called genetic balance. So here in this thing, if this ratio, X by A ratio, X chromosome number by autosomes set number. So if the autosomes are 2, that means in diploid organisms, if 3 X chromosomes are there, it is 3 by 2, it is equal to 1.5. So like this X chromosomes number by autosome set ratio decides the sex of the individuals. So if these ratios, they fall between 1.5 and 1, so 1.5 and 1 above. So 1.01 to 1.5, such type of things develop into meta females. are hyper females. And if this value is 1, they will develop into normal females. And if this value is less than 1, then they will develop into either intersex or normal males. If they are exactly zero point 0.5, they develop into normal males. If they are in between zero point 0.5 and 1, they will develop into intersex or hermaphrodites. And if they are lesser than zero point 0.5, then they will develop into meta males. So here in all these cases, normal male, to become a normal male, a single X chromosome is not required in these organisms. Sorry, a single Y chromosome is not required in these organisms. Why? Because even though Y chromosome is not there, they can give normal male also. So why? Because to give a normal male, the embryo should possess one X chromosome and two autosomes ratio. So if two autosomes are there, that means if it is a diploid and it is possessing one X chromosome, that is enough to produce that type of embryo into a normal male. Y chromosome is not required. Whereas in human beings, Y chromosome is very, very essential. Why? Because if Y chromosome is not there, SRY gene also will not be there. If SRY gene is not there, that that factor will not be produced. So that what happens, the embryo cannot develop testes. So that's why having Y chromosome is important to produce a normal male. But in these organisms, it is not. Y chromosome is not important. If that ratio is 0 0.5, then this can become a normal male. So in some rare cases, if two X chromosomes are there, also that can give rise to normal male. In such cases, the organism should be a tetraploid. 2 by 4, sorry, 2 by 4 also produces what this 0.5 ratio. So such type of tetraploids which are having two X chromosomes also can give rise to normal male. So in this way, if the ratio is exactly 1, they will become normal females. If the ratio is exactly 0.5, they will become normal males. If the ratio falls in between 0.1 and 0.5, they become hermaphrodites or intersexes. So in these organisms, half portion appear like male and half portion appears like female. And if the ratio is above 1, they become meta males. If the ratio is below 0.5, they become meta males. So in this way, the ratio of the X chromosomes to this autosomal set decides the sex of this Drosophila organism. This is called genetic balance mechanism. And third mechanism is male haploidy or haplodiploidy mechanism. This also can be seen in some insects like bees, ants, sawflies, and wasps. In these organisms, if the eggs are fertilized, they will develop into diploid males. If the eggs are not fertilized, those unfertilized eggs are haploid and they will develop into male. So here the sex of the individuals is decided by the whether it is a haploid egg or diploid egg. Haploid egg develop into males and diploid eggs develop into females. So in these organisms, meiosis is normal in females, but crossing over and reduction divisions, they will fail to occur during spermatogenesis in males due to this haploidy. And in these organisms, for example, let us see the example of honeybee. In these honeybees, they will be possessing 32 chromosomes, and egg can produce two types of eggs. So if egg is fertilized, it will have 32 chromosomes. And if the egg is not fertilized, it will possess only 16 chromosomes. The male that possesses 16 chromosomes, the egg that possesses 16 chromosomes will develop into a male organism. And diploid zygotes, they can differentiate into workers or queens depending on the necessity of the colony. So in this way, the fertilized eggs develop into females and fertilized eggs develop into males. And last one in this genetic system is a single gene control of sex. In some organism, only single genes are found that are responsible for the determination of sex. Examples are Chlamydomonas, Neurospora, yeast, asparagus, and maize in plants and Drosophila in animals. Coming to maize, in maize we can find A tassel, where this is a male flower, and silk, this is female flower. And two genes are responsible for this tassel and silk. TS is responsible for tassel, and SK gene is responsible for silk. So in some genotypes, both SK genes will be there. In such cases, they will be having both female flowers only in single plant. And if the plant is having two TS genes, then it will possess two male flowers only, but not the female flower. Usually, these maize plants, they are hermaphrodites. That means each each maize plant will have one male flower or tassel and another male female flower silk. But in some cases where the both genes are the same, like SK or TS, either they will possess both the female flowers or both male flowers. So in this way, the single gene will be there. So when SK gene and TS gene are present in the same individual, they will develop into normal maize plants, which will possess a tassel and a silk. And in Drosophila also, TRA gene is there. This is called transformer gene. So in homozygous condition, when both TRA genes are present in the female, that female fly will be converted into a sterile male when both TRA genes are present. But it will not happen in case of normal male individuals. If the male is having both TRA genes, it will not converted, or it will not transform into female. So therefore, XX female with both TRA genes will be a sterile female, whereas both XX male with TRA genotype will still a normal male. And second type is metabolically conformation of metabolically controlled sex determination mechanism. So one scientist named Riddle, he found that metabolism has some role in the determination of sex in pigeons and doves. So when the metabolism of these organisms is increased, that increased metabolism resulted in the development of male potency. When the metabolism is decreased, that resulted in the femaleness in the embryos. So when the embryos are developing, the metabolism rate decides the sex determination in these pigeons and doves. And A.F. Schull and D.D. Whitney, these two scientists, they found that metabolic rate in rotifer animals decides the male individuals than the female individuals. So if the metabolism rate is increased, more male individuals will develop in these rotifers, and if it is decreased, less female individuals will be sorry. If metabolism rate is decreased, more females individuals will be increased in this rotifers. And next one is environmentally controlled sex determination mechanism. This is best seen in many reptiles like turtles, alligators, crocodiles. So they will lay eggs on the usually seashores or river banks. Usually they dig pits in the sand, and they will lay eggs in the sand. So the temperature of that area decides the gender of the offspring. If the temperature is high, like 30 to 35 degrees centigrade, those eggs will develop into females. The temperature is low, like 23 to 28 degrees centigrade, such type of eggs develop into males. And likewise, some marine annelid, Ophiotrocha, when it is young, it will produce sperms like a male. As it grows into older one, then it lays eggs. So as a young, it acts like a male one, and as a older one, it becomes a female. When some parts of the grown parts are removed from these females, then again it is reverted into male. So in these organisms, the increased size of this organism is converting a male into a female. And hormonally controlled sex determination mechanism. And in this type, sex determination in some organisms like Bonellia will be decided by some hormonal effects. So in this Bonellia, once the larvae are released from the mother, all these larvae, they will freely swim in the water, and at that time they are not sexually differentiated. But the free individuals, that larvae that freely swim and that live freely, such type of larvae, they will develop into females. And some larvae, they choose a parasitic life, and they will attach to the females, usually proboscis of the female. Such type of parasitic larvae develop into a male. So in this way, these female larvae, they will produce some hormones. Those hormones induce the development of testes in these organisms, or development of male gonads in this larvae. That's why this larva develops into female. So when the hormones of female is there, the hormonal effect decides the larva to develop into a male. So in this way, hormones are having a role in the sex determination in Bonillia sea worms. And another example is free Martin. In case of usually cattle, we can see in some cases the cattle can give birth to twins. So if both the twins are females or both the twins are males, no problem will be there. But when the twins are, one is female and one is male, in such cases, usually females will be sterile, only males will be fertile. Why? Because when these female and male twins are developing in the mother's womb, the male hormone effect suppresses the development of ovaries in this female. So that's why these females will become sterile. So this type of twins are called as free Martins. So here also female is becoming sterile because of the hormonal effect of male twin. So this is about sexual determination methods in animals. In next video, let us meet with another topic.